Hi, everyone, and welcome to today's CIC lecture. So it's my honor to present Dr. Maxime Descoteaux, who has graciously accepted to give this lecture today. So Dr. Descoteaux is a professor in computer science since 2009 at the Science Faculty of Sherbrooke University. He is a founder and director of the Sherbrooke Connectivity Imaging Laboratory, or the CIL, where there are about 15 to 20 graduate and medical students and postdocs. His research focuses on brain connectivity from state-of-the-art diffusion MRI and multimodality acquisition, reconstruction, tractography, processing, and visualization. The aim of the CIL is to better understand structural connectivity, develop novel tractography algorithms, validate them, and use them for human brain mappings and connectomic applications. Dr. Descoteaux did a postdoctoral fellow at Neurospin under the supervision of Cyril Poupon and Denis Lebihan. He also obtained a PhD in computer science at INRIA Sophia Antipolis Mediterranee, supervised by R. Derish, after he obtained an MSc from under the supervision of K. Siddiqui in computer science at the Center for Intelligent Machines, McGill University, where he also obtained a BSc, graduating from the Joint Honors Mathematics and Computer Science program. Dr. Descoteaux holds the U. Sherbrooke Institutional Research Chair in Neuroinformatics. He has been cited over 9,700 times and has over 135 journal publications, according to Google Scholar. So thanks again for accepting to participate today. And as a reminder for everyone, you can post any questions you have in the chat or raise your hand. And Max, I don't know what you prefer. Do I interrupt you if there's a question, or do you prefer to keep them for the end?
Yeah, maybe it's easier at the end.
Sounds good. So you can post your questions in the chat then, and at the end, we'll have time for a period of questions. So thanks again, and the floor is yours.
Okay, thanks. Thanks for the introduction. Happy to be here with you guys today. This talk is mostly a walk through some of the basics of diffusion MRI, but also some of the challenges and exciting research going on. And I'll finish this talk with one application in Alzheimer's disease that I hope can stimulate you. I'm already collaborating with some of you here in the room, so happy to open the floor for discussion. And I think I'm online an extra half hour after to answer student questions. So, all right. So I like to start my talks with this quote from Marcellus Musalem, a very famous neurologist that used to be at Harvard, now at Chicago. It says, "Nothing better defines the function of a neuron than its connections." And so another way to say this is, if you know how the brain is connected, you may have a very good window into how it's functioning. And I like to joke about this a little bit. So when you open Google Map and you ask to go to the SAQ, well, you know, Google, just by knowing where you're going, knows what you're going to be doing tonight and drinking a bottle of wine or having a party. And so it's a bit the analogy. So to better understand the brain, we need to understand the structure and the connections. And that all goes through white matter. And so a little bit of biology and anatomy to start with. White matter has its very unique environment. It's very complex. It's rich at the microstructure level here. So we're really here at the scale of microns, where we can have myelinated axons in blue, unmyelinated axons, and loads of cells around them that support their health. So astrocytes, oligodendrocytes for maintaining the myelin sheath, and microglia as well as blood vessels. So it's not only about axons, but a lot of different cells around these axons. And again, let me stress this, this is white matter at the microstructural level. And then, of course, the holy grail of white matter imaging would be to come up with biomarkers, quantitative markers that can detect if this is a healthy bundle of axon with healthy myelin and a healthy cell environment around the myelin. It'd be amazing to be able to detect these anomalies of the white matter. That is, if you're losing myelin and you're in a demyelination scenario, if you're losing axons, you're in an axonal loss scenario. If our axons are swelling or disrupted, you'd be able to capture it as well. Or if you're in a scenario of neuroinflammation where you have an activation of microglia cells, here you see a little bit more red or more activated red cells here, as well as an increase in extracellular water, you want to have markers sensitive to this at the microstructure level, so at the pixel or the voxel level. And so that's the holy grail, and I'll try to come back to this at the end of my talk. Then, of course, the unique feature also of white matter is that there is a macrostructure organization of white matter. Axons microstructurally tend to organize themselves in families, or what we call bundles of axons or fascicles. And here, with a bit of Photoshop, I can show you in color different families of connections of axons for the visual system, for the motor system, for the... And so we can start asking the question, not just microstructurally what's happening, but also macroscopically, where is these phenomenon happening? And of course, this is the relationship with function, because these white matter connections connect the neurons at the cortical level or subcortical level. So keep that in mind all through my presentation, that there's this microstructure feature of the white matter as well as the macrostructure feature of the white matter. And one of the biggest challenges for us today is to bridge this gap between micro and macrostructure with novel imaging techniques. Also, this number is quite huge and scary, but we always hear about the number of neurons, up to 85 million neurons is estimated in the brain, but this number is also quite scary, where we have up to or around 100,000 miles of axons, or 160,000 miles of kilometers of axons crammed into our head. So how are we going to be assessing this quantitatively? Today, most of my talk is going to be about diffusion and diffusion MRI, although I'm going to talk a little bit about multimodality, maybe in myelin imaging or vascular imaging, but most of the talk is focused about diffusion. And so why diffusion? Because it gives us a unique window into the microstructure and the macrostructure. That is, here, if you look at these little drawings, diffusion will be sensitive to water molecules diffusing around axons in their extracellular space. It'll be sensitive to water molecules trapped inside the axons, diffusing in the tubes or in the axons. And we will be able to extract these orientation and reconstruct macroscopically what's going on. And so it's kind of our virtual biopsy or virtual microscope here that allows us to zoom into the microstructure and be sensitive to different water pools. Water inside, outside axons, water that's freely diffusion versus water that's restricted. And of course, if you start having anomaly in your tissue, well, the water diffusion will be sensitive to this anomaly, and hopefully we can pick it up as being abnormal or deviating from normal diffusion. And so it's using our good old MRI scanner. It's just that we're gonna, a bit like the Google cars driving down our streets and taking tons of snapshots to reconstruct the 3D view of Street View. We use diffusion MRI to measure different orientations and have different signatures of the diffusion of the water molecule to reconstruct the full phenomenon. So here is what diffusion is. Diffusion is just random walk. It's quite old. Robert Brown, who was the botanist, described this as Brownian motion. So without obstacles, these water molecules are just freely diffusing around until they hit the boundaries of the box here. And what I want to say here, I don't want to teach you or give you a course about diffusion, but Albert Einstein was the first to write the equations of free diffusion here. And the bottom line that I want to teach you today is that you can quantify diffusion here with the apparent diffusion coefficient. This is a speed. This is basically telling you at what speeds are the water molecule diffusing here. And so if you do a Wikipedia search about apparent diffusion coefficient, you have an apparent diffusion coefficient table of basically blood, water, aqueous water, any type of medium has its specific apparent diffusion coefficient. This is quantitative and characterizes how quickly water molecules diffuse when they're free. So of course, this becomes more complicated when they're restricted, but keep that picture in mind about apparent diffusion coefficient. And let me just give a little Google map picture of this. Imagine you're doing a party in front of McGill in the quad, and you're having a party, and this is free diffusion, just people randomly talking to each other, having a good time. And basically, you can see it just that looking at how people move, you have an idea of what's the structure underneath. So if you move the party into the street, you see that, of course, as people party in the street, they start molding the structure or the shape of the street. And if you move the party into an intersection, well, again, at the beginning, it looks like isotropic or free diffusion, but after a certain time, if you let diffusion do its process, you can see a very nice crossing configuration showing up. So I think this serves as a very powerful example of the power of diffusion, the power of letting the water molecule explore the structure and leave a signature. And in principle, if you let the water molecule diffuse for a long time, enough time, and if you have the proper signal from MRI or from anything, you can most likely assess indirectly the underlying structure. And this was discovered by Denis Le Bihan in this famous paper, Diffusion Weighted Imaging in French, in the French Academy of Science, who basically said that this apparent diffusion coefficient can be measured from images. And just so people don't all know this, but the famous B value is because of Bihan, Le Bihan here. And so he was my postdoc advisor. And this famous paper just nicely shows that you can measure, c'est possible de calculer le coefficient de diffusion moléculaire. You can measure this apparent diffusion coefficient in brain tissue. And that's now, very old, almost 40 years old. And what Denis Le Bihan showed is that if we acquire enough data, we can have an idea of what's normal apparent diffusion coefficient. And so that if you have a tumor, a stroke, a cancer, or something abnormal in your head, of course, this apparent diffusion coefficient is going to deviate from normality. And this is what made diffusion on the map very quickly. It was really quickly adopted by the clinical community. And one of the first killer application of diffusion weighted imaging was stroke, 1992. Super fast acquisition, 1 to 2 minutes. You just acquire a few images, minimal of 3 images at different B values or 3 different orientations, and you can have more information than your basic T2 weighted imaging. So I'm not going to explain all the physics of this, but basically a stroke can appear on the DWI image or on the apparent diffusion coefficient image right away, right after the incident, as opposed to in the typical T2 weighted image after a few days up to a week. And even today, this is probably the success story of diffusion. It is the most useful clinical application, and in a sense, this was mission accomplished for diffusion. And that's why if you do a keyword analysis of papers in PubMed, and this is a 2002 search, right away what you see are quite clinical words: ischemia, stroke, infarcts. And so diffusion, right after its invention, was really quickly adopted by the clinic, patients. And so you don't see the typical jargon of tractography, tensors, and whatnot. This will come, but I just want to emphasize that diffusion is used in the clinic with its fair load of success, especially in stroke. Sorry. But what we quickly realized is we need more than that. We need more the apparent diffusion coefficient. We need to know where and how the white matter is organized. And I just want to reiterate this if people are not familiar with these images. This is a T1 weighted image, a structural image where we see the white matter and the gray matter in gray. And this is a myelin map. It's much more recent, but all I want to say here basically is that white matter is just one contrast, right? It's white. Where is the structure there? Where do I see my motor tracks or my arcuate tracks? No matter what the orientation is of the axons underneath, we still have a signal that's just white. It's one gray value. And so what researchers in the field quickly realized is that we needed something sensitive to orientations if we truly wanted to map the directions of axons, not only the apparent diffusion coefficient. And so just to illustrate this, if I take, again, my MRI machine, and I acquire 3 images, one in the X direction in red, one in the Y direction, and one in the Z direction, this is the raw signal from a human Connectome project data set. And I just want to emphasize that the signal is completely different depending on the orientation you measure it. Right? This is not like a typical structural T2 weighted or T1 weighted image. So for instance, here in the motor track, when you measure in blue, it's dark. Whereas when you measure in X, it's bright. And if you measure in Y, it's bright. And so there's this clearly nice, sexy mathematical engineering problem that needs to say, okay, there's this nice inverse problem that we need to acquire a bunch of images in space to extract orientations of diffusion and then quantify apparent diffusion coefficient. And probably most of you know this word DTI. This was really one of the first revolution to map orientation was diffusion tensor imaging, where Peter Basser, 1994, showed that with a minimal of 6 orientations in space, you could put that into an inverse problem and reconstruct at every voxel how does the water molecule diffuse orientationally. So not just have a number that describes the apparent diffusion coefficient, but now as the principal direction, how is diffusion along the main direction, how is diffusion across the main direction, and start being a bit more sensitive to the actual microstructure. So this is DTI in a nutshell. It's been cited more than 10,000 times now, and it's a relatively standard now, fast acquisition. And this was the first time that we could show in 3D the white matter architecture. We could create maps of how is anisotropy, where are the most structured roads that have a diffusion tensor with a profile of FA1, and where is diffusion most isotropic? So where is diffusion freely diffusing? A little bit like my introduction with my Google Map video. And so, this gave birth to the DTI era, I would say, which in some respects we're still in, because it's DTI this, DTI that. In fact, for most people, DTI is a synonym of diffusion MRI. So I always try to tell people, DTI is just a model. It's just one way to deal with diffusion data. And so this is more and more recognized out there. We can do much more than DTI, but there was clearly the phase, and we're still in it, of I love DTI, where MRI vendors were putting DTI everywhere instead of diffusion MRI. We saw the tensor appearing, and basically DTI being applied to every potential brain disease where advanced imaging was used. So why is DTI still not perfect? Well, the next big challenge that we discovered is that we had crossing fibers and partial volume a bit everywhere in the white matter. And this is just a small example here, but if I zoom into a voxel where you have potentially 2 families of axons traversing here the corpus callosum and the motor tracts, you potentially have partial volume with CSF when you're in the border of ventricles or deep structures. You can have gray matter partial volume, and you can throw in there pathology. So it's a highly complex microstructural environment that DTI only tries to summarize with a single diffusion tensor representation. That is just a football or a soccer ball that needs to explain this on average. So you quickly realize, at least theoretically and in terms of engineering, that this will not do it. This will be limited. Doesn't mean it's not useful. Doesn't mean you cannot be sensitive to disease or to abnormality. But it's very hard to know what's going on and to be specific. And just as an illustration here, if you take 4 different kinds of scenarios of white matter microstructure where you have a healthy bundle of axons with super sharp diffusion tensors, if you have a tight crossing fiber, if you have a demyelination scenario where you have water diffusion more freely diffusing perpendicularly, or if you have appearance of inflammation with pockets of free water, well, it's quite easy to show that up to a certain limit, you can always be in a regime where FA is equal, about 0.4, 0.5, and this is basically the average FA in a human brain. And so it's highly non-specific. It's sensitive, but not specific. And tons of papers out there, depending on the pathology. Now I took a review on mild TBI, but maybe half the papers show an increase in FA and the other half show a decrease in FA. So maybe that's an indication that DTI has reached certain limits and that we need to zoom in a little bit more to disentangle the different microstructural phenomena. And so, as a joke, for a while, a lot of people were using FA as a biomarker of white matter integrity, right? You have a disease A, you measure DTI, and you were hoping that FA was reducing with disease. And because it was your marker of white matter integrity. But there are so many scenarios where actually FA can increase with pathology because of contamination with free water, because of crossing fibers, because of several different phenomena. And so we need to be a bit more savvy about what we're measuring, and especially about interpretation, how we interpret DTI results. So, one of the take-home message today is don't use DTI as a synonym of diffusion MRI. It's just one way to look at your data. Because crossing fibers are everywhere. You have, and this is, you'll see a slide next, but you have basically about 2/3 of the white matter, between 2/3 of the white matter to 90% of the white matter at the current imaging resolution of MRI that is thought to be under a crossing fiber voxel. That is pure crossing, sometimes kissing or branching, but nonetheless, 2 families of, or 3 of fiber population coming together inside the same voxel. And so this is a tremendous challenge for diffusion, for DTI, and for the whole community in general still today. Because if you actually look at the occurrence of where single fiber populations are in red, well, basically single fiber populations are only in very specific regions of the brain, mid-sagittal corpus callosum, internal capsule, where all the axons are really parallel, very tightly packed, but basically everywhere else in the brain is 2-way, 3-way crossings. And so this is challenging for interpretation and for white matter analysis in general. But thanks to continuous efforts from the community, at least nowadays, we have, with a bit more than DTI, very robust tools to extract crossing fibers. It's still difficult to quantify what's going on, but at least in terms of orientations, we're becoming more and more solid at extracting all these 60 to 90% of voxels with crossing fibers. And this is thanks to the families of spherical deconvolution techniques. This was part of my PhD, but done independently by several groups. But so basically now we can reconstruct robust fiber reconstruction. So this is the fiber ODF that are robust to crossing fibers. And it's not really more demanding in terms of acquisitions, because most people do their diffusion with 30, 40 directions. And we've been showing that with that much data, you can robustly reconstruct crossing fibers a bit everywhere in the brain. Now, the biggest challenge is to know if things are crossing or branching, but that's another discussion. But at least for mapping white matter, for reconstructing tracks, for the last 20 years, this was a big limitations of DTI, and I can say today that this is not a solved problem, but it's a lesser problem than it used to be. So, which brings me to a nice slide that I think will educate most people in the room here, because it's a bit geeky, but it's a new term that I think you will go to sleep tonight having learned something. But a pixel is a picture element in 2D. A voxel is a volume element in 3D. What my community is trying to push nowadays is for a new term called the Fixel. So normally in the room with people in front of me, I would play around with you guys to try to make you guess what the Fixel is, following the logic here: 2D picture element, 3D volume element. Now, probably people are thinking about it in their head. This is a fiber element now. And I want to emphasize this because I think this is really the future of better mapping quantitatively white matter. That is, if our imaging voxel with MRI in 3D, this XYZ coordinate in your image or NMNI, now you have to imagine that this voxel can have multiple fiber elements going through it, right? Because 90% or 60% of the white matter is crossing fibers. So you have a fiber element for the blue axons here, and you have a fiber element for the yellow axons here in this drawing. And each of these pixels can have their own microstructural environment, their own axon density, their own intracellular water here in blue, their own myelin signature, their own oligodendrocyte signature. And so what our community is trying to push nowadays is really towards pixel-based analysis or track-specific analysis that's able to disentangle the voxel in different tracks or pixels. So, the future of mapping tissue integrity will be at the pixel level. We need to go beyond the voxel. And of course, you can imagine, because most of you have experience with imaging and engineering, this is a serious complication, computationally, to deal with that properly in our analysis tools. Because the holy grail, again, is my virtual biopsy, right? If I take a healthy tissue under the microscope and I stain it for myelin, and this is actually a part of the fornix. If people don't know the fornix, this is the memory tract of the brain that connects the hippocampus. So this is a healthy mouse model sliced up across the axons. And if I draw in different colors the different compartments, my holy grail, right, would be to have a quantitative measure of how much axons in this pixel, how much myelin, that is how much purple, and how much water, extracellular water in this pixel, right? This is my holy grail. If I can have a measurement tool that says how much green, how much purple, how much blue, I can really turn this to a really powerful analysis tool for clinical trials or for clinical research. Take an Alzheimer's tissue here. So this is the same region inside the fornix, but this is a 15-month-old mouse where you can clearly see that, wow, axons have changed in shape and size, and there's some sort of swelling. Axons have been lost in the process. So clearly axon content here has decreased and has changed dramatically. Same with myelin. Myelin has deteriorated, granulated. There's a loss of the myelin sheath and a huge increase in extracellular water. So keep these pictures in mind, but essentially you want to do this now in the Fixel world, right? If I go back here, this is a slice through this guy. So if I can quantify these things, I can really start mapping tissue integrity and having a good interpretation of what's going on, something that was impossible to do with just a diffusion tensor. And so this is where we're at as a field, I would say, so that, you know, we can start capturing healthy axons, crossing fibers, demyelination scenarios, inflammation scenarios, because we can pick up the free water versus the tissue part of the signal. And so these two things are critical to do good fiber tracking, and these two things are critical to do good white matter assessment. And as I'm going to show you now, if I want to go into the Fixel world, I'm going to need fiber tracking to determine what are the fibers going through my voxel and which Fixel should I be quantifying in my analysis. And so this brings me to tractography. So this is the way that we connect the dots and reconstruct from A to B the most likely pathway that the water molecule traverses by following axons. It's a very indirect process. I want to stress here, I'm not tracking axons per se. I'm tracking water, how water diffuses in my images. But this also is quite late, you know, it's an old field. First abstract was in OHBM in 1995. And you can see the quality of the images. It's quite funny. This was from the group of the MGH. So NIH, MGH, and the French group with Le Bihan in parallel came up with the idea of, you know, tracking the orientation in space. And so here's just an illustration from my lab, now that we can use in surgical planning, where we can really track around the tumor, and we can do this in real time. We can move around, explore the edema, explore the necrosis area, and try to have a 3D mapping of what's going on around a brain tumor, for instance. So this is, again, a small application, but an application that we see a bit more in the clinic. So aside from the stroke application, this is probably the second most used application and the only application of tractography, as I know, in the clinical world. So for instance, this is us in Sherbrooke, when we used to go in person preparing the data before the surgery. We used to connect to the neuronavigation system, had a flat screen, and in real time during the surgery, we could link the cursor with the operation tool of the surgeon, and he could explore the tumor and the brain architecture as he was operating or waking up the patient, stimulating different cortical regions. And so this was a very research and development project with the hospital. So this, just to show you the application of tractography, it's not just built to make pretty images, it's actually being used regularly by neurosurgeons. So by 2012, we saw words such as tractography, connectivity appearing in the nomenclature. And this is about when the Human Connectome project appeared as well. And since then, you know, Connectomics tractography is even more present in the cloud, the keyword analysis of research using diffusion MRI. So what's the after tractography? What do we do with tractography? Well, a big challenge computationally was to segment tractography. Just like we use an MRI image like we do in Millar's lab to extract the hippocampus, the different subfields, the different cortical regions, the different nuclei. Well, we have the same problem, but in the world of tracks. We have the problem of defining the biggest highways of the brain because we know them anatomically, we know what they connect. And so it's quite easy nowadays to do this with semi-automatic or fully automatic tools that will dissect virtually a full brain tractography to create these tracks. And once we have these tracks, well, we can do our Fixel-based analysis or do some morphometry on them. So this gave birth to the word Tractometry, which is the act of quantifying tracks. So you can quantify the volume of a track. You can see if there's atrophy in time. You can quantify the Fixel-based fiber density, the Fixel-based myelin index, the Fixel-based inflammation. and accumulate that in your statistics to run your study. So this is called tractometry, or sometimes track profiling. And last but not least is the Connectomics application, where more and more tools are available here now to do structure function mapping, where now you would use an atlas of the cortex and basically extract all the pairs of potential connections to quantify them or to go into network science. So I'm not going to get into this today, but I just want to make sure that, you know, having dedicated tools to do this properly is difficult. To bridge the gap between, the gap between tractography and T1 images, or tractography and surfaces, is actually very challenging. And Etienne Saint-Onge spent most of his thesis doing this, and now he's a postdoc with Louis Collins at McGill. So you might hear about Etienne, and you might be able want to collaborate with him to really use these cutting-edge methods to connect surfaces and meshes with the diffusion world, to really go towards multimodality integration. Because this is where your functional signal lies, this is where you have your cortical thickness, this is where you have your EEG, fMRI, potentially different signals. And so if you want to link it with connectivity and connectomics, dealing with surfaces and with tracks is actually very challenging. And we have tools now to answer these questions. So bottom line, nowadays our cutting edge pipeline uses diffusion, T1s to extract the surface, the different white matter, gray matter, CSF, the parcellation. We deal with crossing fibers properly. We join the parcellation with the tracks to generate the connectome. And now the biggest question is, what do you put in this matrix? It's been quite well documented that putting track count, that is the number of tracks, is not a great quantitative measure. We have alternatives to this, but this is really a challenge and a work in progress in our field. But just so everybody's on the same page, given a parcellation and given a tractogram, you can basically start extracting every pair of connections. We have fancy ways to detect outliers, to clean up the results, to make sure we really have truly anatomical tracks connecting our regions. And then, if you think about it, you can quantify Fixel microstructure to put that number in that matrix. But it quickly becomes a very multidimensional problem where you can have your connectivity matrix for apparent fiber density, so fiber density for myelin, for inflammation, for FA, for volume, for length, for track count. It quickly becomes a very big, high-dimensional data that network scientists now can have really fun playing with. And so then you can see the brain as a multidimensional graph. So I want to make sure I have time to talk about applications of all of this. I chose just a couple. And there's tons of them, and I'd be happy to discuss this in the extra half hour. So the first application is the Disconnectome application. So almost every neurologist or neurosurgeons I know would love to have this app that basically you press a button and you know how and where the brain is disconnected. And I think we're getting closer and closer to this. We have now atlases of the connectome, of what are the most occurrent connections in a population, in a healthy population. So we can ask ourselves, how does a stroke, how does MS, how does a traumatic brain injury disconnect the brain? And I think in the future, even go a step further, not only how it disconnects the network, but quantitatively, are we in a demyelination scenario or a loss of axons or cell swelling or an inflammation scenario? With the connectome as a backbone and tractography and the Fixels as the backbone, we can now pretty much ask these questions. So that if we have an MS, for instance, with tons of lesions that are enhancing and flare, or that are black holes, that are demyelinating, or that are acute or chronic, we can really start having a pathology-weighted connectome. Same with traumatic brain injury, stroke, and a lot of brain diseases. So this is the disconnectivity application, and it's all new. So it's very exciting and lots of things to be done here. The other application I wanted to talk about today, because I know a lot of people at McGill are working on it, is Alzheimer's disease, where it is known through time that you have brain atrophy. You're losing gray matter. There's also a loss of white matter. So this is very macro. Now a more famous marker also is the hippocampal volume. But what about the white matter microstructure? And in fact, there are some very nice, very old papers out there, both in animal models and clinical applications that I've shown clearly that in the AD spectrum, there is a loss of axon, there is a sign of demyelination, there is a loss of oligodendrocytes. So investigating more closely the role of white matter microstructure is definitely something that is now doable and that we've started doing myself in my lab. And so let me come back to this picture. Keep that in mind, but I'm going to try to show you that if you can map these three markers, even though they're indirect and with imperfect technology, you may have a window into white matter microstructure cross-sectionally, but also in time. And this might be a place to look for if you're running a clinical trial or if you have a drug and you want to see if it's remyelinating or it's reducing inflammation. I believe we're almost there, but we're starting to have measures that can help for this. So let me illustrate this with Alzheimer's disease as an application and the ADNI database. First is to ask the where are we going to look in the white matter? And so we have several locations that have been published in the literature in the past 20 years. So there are regions, especially in the temporal lobe, the limbic system. The fornix is not on that list, but I should have put it there. This is just I forgot on my side. But most of these tracks connect the temporal lobe. They end in the temporal, either the medial temporal lobe or the whole temporal lobe, some of the limbic system. And you can wander there and you may want to have a look at these papers. How is white matter microstructure there changing? And what is the link with PET, for instance, in this paper? Very nice paper linking PET, FDG, and ketones with these white matter tracks. So I think we have a virtual dissection / biopsy tool that can ask the question where and how. The where is my tractography and my track dissection. And the how is my white matter microstructure indices in the Fixels. So if I have a map of extracellular water, fiber density, and myelin markers, I can map it on my tractography and study how this thing changes in time or after a certain intervention. So very small phase 1b trial, just to illustrate my point, and I'll conclude with that. And I use the ADNI longitudinal data as a reference, just to show you what type of things we can measure from a diffusion MRI. So first is our indirect indication of extracellular water, right? The amount of blue here, which we saw in our histology scenario. And we had a healthy tissue and this histology slide. The black curve here in gray is the ADNI reference over 24 months with 36 patients. So this is the typical evolution of white matter in the Alzheimer's disease bundles here over 2 years. A slight increase, like a 12% increase in extracellular water. A little bit like we see in this picture. Where with this new anti-inflammatory drug of the recent years, it's called Expro. I don't want to get into the biology of it, but it's a good anti-inflammatory. You actually see what's happening after a year. These are not true stats. I give you that. This is just with 653 patients. This was a phase 1b to assess safety and dosage. But nonetheless, we can see that there's a clear trend that something is happening, reducing the amount of extracellular water. Now, what about apparent fiber density? The amount of green here. Again, with ANI, we see a slight decrease in time of fiber density. Yet with this drug, after 12 months and in time, we see an increase in fiber density. Something that could be tempted to interpret as some sort of tissue repair or strengthening of axons. Nothing is validated here. I'm just showing you the types of things you can see and start digging into. And last but not least, an indirect myelin index. You can see with ANI, you have a sort of demyelination, slight demyelination with time, which is indirectly linked with radial diffusivity. We want to confirm this with a truly specific myelin contrast, but this is, like I said, just using diffusion. And again, with XPro, we see an improvement in myelin, not meaning that we cannot know for sure that it's remyelination, but, and I don't want to oversell this technology, but if you deal with crossing fibers, if you deal with partial volume, if you're in the Fixel world, chances are that you're getting closer and closer to the specificity of the white matter microstructure. So this was just a small illustration of the potential power of all these tools. And so I want to conclude by just coming back to my initial quote. If you remember, my initial quote was, nothing better defines the function of a neuron than its connection. I want to go a little bit further today and say maybe nothing better defines the function or dysfunction of a neuron than its connection, true, but also its integrity. And I think this in the future will not come just from diffusion, but I think we will soon have Fixel-based myelin contrasts. We will have vasculature. We may have something related to oligodendrocytes and glia cells. And I personally think in terms of computational backbone, tractography and white matter might be the perfect tool to combine all these modalities together. So with that, I'll leave you with a few futuristic notes about Connectomics, that we will need a revolution in Connectomics if we want to discover more about the brain. We'll have to measure it quicker, decode it, compare it, change it in time, if we want to really have an impact. So thanks again for having me, and I'd be happy to answer any questions. Yeah, I went a bit overboard, but sorry about that.
I mean, it was so interesting. We didn't see the time pass. So on that, then, yeah, we'll open the floor for questions. So you can either type your questions in the chat. You can raise your hand on Zoom if you want to answer out loud. And I see we already have a question. So Nadia, you can go ahead if you want.
I was wondering why they started looking at, okay, maybe I'll type it at strokes first. Like, where did they decide to examine, like, why did they come up with this theory that people in strokes would have altered, diffusion weighted image acquisition?
So let me try to make sure I understood. But so the frustrating thing for a clinician about stroke was that they had to wait 3 days, more or less, to see something in MRI. With diffusion, because of the stroke, because it affected how water molecules were diffusing locally, depending on if apparent diffusion coefficient was increasing or decreasing, they had a totally different branch of diagnostic charge at the hospital. So I think if you're reducing, so if you have a black ADC, you're more in trouble than if you have an enhancing ADC. So I'm not a doctor. Maybe I could get some help here. But so it really changed the pathway or the pipeline of patient care in the hospital right at the acute stage when the patient comes out of the ambulance. So that was the killer application because, and to be honest, I think even with mouse model, they still don't fully understand what's actually doing in the tissue that's affecting the water molecule diffusion. Is it the permeability of axon? Is it swelling? Is it just like inflammation or a little bit of all of the above? It's still an open question in our field to know what exactly is happening. But it's a little bit like if you take your kid and he has a fever, right? You don't know what's happening, but you know he has an infection probably, and something's going to be happening, even though you don't know specifically what's going on. So it's kind of it's a thermometer of the year 1990 for stroke.
Right. So I don't see a question right now, so I think I'll jump in with one of my own, but in the meantime, feel free to add more to the chat or raise your hands. One thing you show in your presentation is that initially we started with really diffusion. It was really like hard to see pictures, and then we zoom in with DTI where we start to see the fibers, but it's not really precise. Then we zoom in with the fiber direction and other metrics in the fibers and the Fixels, as you call. So how much more do you think we can zoom in? Do you think one day we'll be able to go intracellular, see the water, how it diffuses within a neuron, or where do you see the advances towards that?
Yeah, it's a great question and a bit about future, but a lot of interest now is about bridging the resolution gap between MRI and non-MRI methods. So there's a quite strong expertise in Montreal between the Polytechnique and UQAM about microstructure, microscopy, polarized light imaging, OCT, everything based on the optics, where you can really go down to the neuron and single axon and learn about, for instance, I'm getting into this world, but axons can make collaterals. So a single axon from a single neuron can actually project to 100 sites. So if you think about my talk today, this is nowhere addressed, and it will soon be a nightmare. But so right now I'm reinventing a bit my algorithms to deal with this complexity, that now if I have a big, microscopy mosaic of a mouse brain, and the axon is allowed to branch and make collaterals. So with one seed, you make, 100 connections. It changes everything computationally and in terms of connectomics. But it's at a scale that is nowhere near what we can see or quantify with MRI. So right now I have a feeling that there will be efforts from MRI better mapping microstructure, microscopy better mapping microstructure, and then the two fields will have to talk to one another, and maybe with some of the AI tools out there that are available now, maybe we're going to be able to complement and augment our MRI images given some priors from microscopy. But this is a bit futuristic. But yes, eventually we would want to have single axon, single neuron synapse mapping. But this is, I highly recommend the book, actually, Connectomics, by Sebastian Chung. It's a great TED talk as well, but he clearly describes the challenge of human connectome mapping at different scales. So the micro, the meso, and the macro scale. And everything I showed you today, even though I talk about microstructure, it's really macro, right? It's the voxel, it's the MRI voxel. So lots to do. And it's a computational problem, because it's a nightmare to deal with these images and to segment and track and merge different field of views to create just one mouse brain at this resolution. It is it's an incredible tour de force.
Yeah, no, I was gonna say we'll probably need better computers to do this. So thank you for your answer. Malard, I think you have a question.
Yeah, thanks. Great talk, Max, as always. Thanks a lot for doing this.
Thanks.
I have maybe a question that's in between what you showed today and what was just asked, and that is, most of the tractography that we use, and then as a result, most of the quantification that we do is really in the major fascicles, right? Which really actually only represents a very small proportion of the total number of axons in the human brain. So what are your thoughts on using a lot of the strategies or any of the strategies that you mentioned today in kind of the stuff that doesn't have names, right? Because we tend to go for the stuff that we can name, which are the fascicles, but all these U fibers, for example, between cortex, and then you mentioned some of the Tensor that kind of takes care of that blade problem. What are your thoughts on, are we at a level where we can even say that, can we get to those U fibers that probably have, which are probably the first things to go before the fascicles go in terms of, say AD or aging or something like that. So do you have any thoughts on that?
Yeah, I do. And so just to make sure, when we talk about U fibers, they're really U fibers between one gyrus and the other. So that's the first thing, because we can have small, very short tracks within the gyrus. This we're not ready for.
Yeah.
And this is something that's kind of too short and ignored in everything we do. But to have the gyrus to gyrus U fibers, I think we are ready for it. But like I like to say, they need dedicated parameters. So I hope one day, because you guys are users of public tools, but we should, as developers of Tractography, provide maybe profiles of parameters that best track U fibers, or that best track projection fibers, or that best track commissural fibers. I think we're also there. I didn't talk about this today, but in my lab, I have students really dedicated to what we call in my world bundle specific tracking. To track a U fibers is not the same problem as tracking the cortical spinal tract, or even tracking the putamen to substantia nigra. So we need dedicated parameters and dedicated algorithms for different brain locations. But I think if you just tweak a little bit our tools, the U fibers, I think you're ready to look at them. One place we're not ready is all the connectivity around deep structures. This is a nightmare. This is a soup of partial volume and little funnels of white matter, and probably huge number of collaterals, and everything's connected with everything, and it's just, I think it's a mess for Tractography. So just as a word of caution, now, when you want to investigate the less unknown tracks, one of the approaches to do Connectomics, right, is you map a parcellation and you wonder how are every parcels connected to one another. The danger there is that I think our tools are providing too many false positives still. It's easier to get lost for Tractography. So my recommendation here is try to leverage group analysis and say, okay, I'm going to study tracks that are outside the well-known fiber tracks of anatomy, but I'm going to make sure they're always there in everybody, or they're there in 90% of my cohort. So it doesn't mean it's not a false positive, but if it is a false positive, it's a feature of the data. It's always there.
It almost seems like you have to do stuff that we were doing, at the early days, saying, "Oh, look, there's a track here, and I can get it reliably," right? Like it's almost a new anatomy experiment every time, which is reasonable, right? Yeah. Thanks again, Max.
You're welcome.
So we'll take maybe I see another hand raised. We'll take maybe that one as the last question, and then we'll move on to the student discussion part. So Jeremy, you can go ahead and ask your question.
Hi, Professor Descoteaux. Thank you for the talk. Nice one as usual. I was just wondering, your last slide about multimodality partly answered my question. It was we have other techniques that can also inform us about white matter, like myelin content or stuff like that. And you mentioned that this multimodality can bring the field forward. Is this something that is already done? And if so, where are we at as of now to combine other modality with tractography or other diffusion techniques?
Yeah, great question. Right now, I would say it's done in silos, but it's starting to be done. But that is, I'm going to collaborate with a group that does the best myelin mapping on Earth, I do my best tractography, but we do things separately, and then we merge the two. This is not my vision of the future. My vision of the future is that orientation impacts myelin. So knowing how many tracks you have in the voxels should change the way you reconstruct your myelin contrast. I think one day we will have Fixel-based myelin imaging, because it's known that, you know, edema, free water, different crossing configuration does affect the actual myelin reconstruction number that you get. Same potentially with other mechanisms that are sensitive to maybe perfusion or vasculature or iron content. So to answer your question, today, everything is done separately. You know, I do my QSM, I do my whatever, and then I map it together, and I try to do little more as post-processing. I have a vision of the future where, you know, MR physicists will try to do this in a unifying acquisition scheme. A bit like, if you know Christine Tardif, she tries to do this for myelin, where it's an inversion recovery diffusion sequence that needs fiber orientations to do the reconstruction. So I think that's, if I was in physics or MR physics, I would be attracted by such a challenge.
Great. Yeah. Thank you.
All right, so we'll stop here for the questions. I think we'll move on to the student discussion part. And I mean, of course, everyone is welcome to stay, welcome to come and ask questions. So it's more of an informal type, but everyone is welcome. So thanks again for the presentation.